Hi Deepa. Hello. Hello.
Good evening.
Deepa sir will be sharing the PPT. CVP.
Okay, ma'am. Okay, ma'am.
I have made sir is present.
Okay.
Chinu sir is. Good afternoon, sir. Was here. Off, I think.
Good afternoon, Dr. Mahalakshmi.
How are you, sir?
Fine. How are you?
I'm
They all
okay.
fine?
Yeah, everybody is fine, sir. How's madam?
Yeah, she is fine. She went to Japan today and came. Yeah.
Okay.
Ma'am, good afternoon, ma'am. My voice is clear, ma'am. Good afternoon, ma'am. My voice is clear, ma'am.
Yeah, little bit echo is there, Deepa. Where is your headphone?
Yeah, ma'am. I'll connect with headphone, ma'am. Means I will connect with I have connected through laptop. I'll connect headphone, ma'am.
It's echoing, Deepa.
Okay.
Good afternoon, Dr. Roy.
Good afternoon, madam. Am I audible?
Yeah, you're audible. I'm Dr. Mahalakshmi. I had written to
Fine.
you the emails. Hi, sir. Deepa, there is some echoing. Or the voice is clear. Hello? Deepa, I can't hear you. I can't hear you, Deepa. Your audio is not enabled. Your headphone is on, but audio not enabled.
Okay.
Now it is.
You are able to hear me, ma'am?
Yeah, I can hear. Is there an echo in the line, Deepa?
No, ma'am. Actually, I thought there is some network problem with this laptop. Otherwise, I thought I'll connect with my wired connection.
There's some disturbance I'm hearing.
But your voice is clear to me, ma'am. Sir's voice, speaker sir voice was also clear to me.
Okay. Hearing some Some background disturbance.
Good afternoon.
Good afternoon, sir.
Good
Your
afternoon, sir.
Good
internet
afternoon.
became okay, sir.
Which one?
Your internet seems to be back. You are now in your
Yesterday
desktop.
afternoon, I think they must have been working something. Till today
Okay.
morning, it was not working. So,
Oh. Yesterday, then this,
I joined by my mobile on.
yeah, this we had this issue, no, sir. So we sent mail to Cisco and then tech support. Then we got a call back from their German headquarters at 11:30 in the night, saying that your problem has been rectified.
See, they are very prompt.
Yeah, they're quite prompt. But anyway, I wish they hadn't called at 11:30 in the night.
Yeah, they don't pick up the phone after 6 o'clock or something.
Anyway.
Good afternoon, Dr. Bhatt.
Good afternoon. Good afternoon.
Oh, sir is here.
How are you?
I know sir is here.
I am fine. What about you?
Yeah, fine. Very well.
Actually, yesterday night it was raining. Electricity was not there. I don't know other
That
plans.
is going and coming. Repeatedly, it was going and coming.
Ravi Shankar has joined. Good afternoon, sir.
Good afternoon, madam.
Good afternoon.
Good afternoon,
Good
Professor
afternoon,
Bhatt.
sir.
Good afternoon, Deepa. So Dr. Subarna has joined, yes. Good
Yeah,
afternoon,
he
Dr.
came
Subarna.
for some time and then his video is off.
He had joined.
Roy, you want to
Good
share
afternoon,
your
sir.
slides and
Can you hear, sir? Yeah. Chal. Good afternoon to you.
We are not
Yeah.
able to see.
Dr. Roy, do you want to share your slides and check? Hello?
Am I audible, sir? Am
Yeah,
I
you
audible?
are audible, sir. You are audible.
Audible but not visible.
Sir.
Dr. Bhatt, incidentally, you know him, is it?
Listen, sir.
Yeah. Now I think it is visible.
No, you are
You
not
know-
visible, sir. Your camera is off. We can't see you.
You asked me something, sir?
Yeah, Dr. Bhatt, you know Subarna Roy?
No, I have not seen him.
You know him earlier. That's what I'm asking.
I saw his picture in that poster.
Oh, okay. सुवर्णा तेरा कैमरा ऑन काम करो ना There seems there is some difficulty.
Ah. Sir, if you can send the slides, we
madam.
can also share if there is a difficulty. The audio is on, but video is off. Then you will call him, sir?
Yeah,
Dean said you are going to call him?
I'll call him.
Okay, sir.
Send Durga. His, his number is
Dr. Roy has come back to the meeting. Hello, sir.
Am I audible, madam?
Ah, yeah, you're audible, sir. You're audible, visible. Ah.
Yeah. Okay.
Would you like to try sharing your slides, sir?
Yeah.
Okay, I'll make you
Srinivasan ji,
presenter.
can you come to eco room little bit urgently required? Okay.
Yeah, I have made you presenter, sir. You can try
Yeah.
sharing, sir. You can run a check before program
Yeah.
starts.
Is it being shared?
Ah, yeah, we are seeing the slide, but it is in normal mode, not in
Yeah.
slideshow.
Fine. Yeah.
Yes,
Okay,
sir.
Now it is okay, no?
Yes.
okay, sir. Sir,
Yeah.
your audio, some echo is there, sir. Can you use a headphone?
Yeah. Okay. Just a minute.
Okay.
Give me a minute. I'll just change the connection.
Okay, sir.
This presentation. Yeah. So. Little bit was I'm in Wi-Fi mode, so now can it be into this? But mine is only USB.
So you want to do with your laptop?
Here also no problem, but this is WebEx. WebEx. Shall I give the? I'll give you that idea.
Oh.
It is meeting number 166. Yeah.
Sir, we have
Yeah,
received one mail from YouTube. Dean sir, if you reach a certain number of subscriptions, no sir, we will get YouTube silver button, gold button like that and all, play button. So we are nearing the silver button rank. So they have actually sent one mail to us saying that
No, what will be this one? What suppose you get a silver button? Any privilege?
nothing, look, it's only a privilege, that's all. You can monetize after sometimes advertisements can be put in your channel and things like that.
Okay. That means your credibility is you have uploaded so many things.
Yeah. And so many people are viewing it and subscribing to it.
Do they have to make payment for seeing or free?
No, sir. They are free only. YouTube will pay us, the creators, money if we have so many followers and subscribers to our channel. YouTube will make money out of advertisements and they will give us little bit. Something like that.
We have paid subscription, no?
Eh?
We have a paid
We have
subscription
taken the
to
G
YouTube.
Suite subscriptions, not only YouTube.
Ah,
The entire G Suite, we have the educational subscription. So that is for all services provided by Google.
oh, so under that G Suite this comes.
Yes, that next to your email, no, sir. The 9 buttons will be there, no.
Yes.
So in that, all that is free. That Google Workplace,
Oh,
it is called now. Earlier, it used to be called
Hmm.
G Suite. So if you buy that license, everything you will get. Google Forms, YouTube, Google Drive, all comes under the same thing. Sir, 3
madam,
o'clock, sir.
I'm shifting to another computer. That will be better.
Okay, sir.
Okay. Fine.
Yeah.
Go ahead.
Dean sir, 3 o'clock, you want to start? Deepa will be hosting.
Well, let him. One minute. You give time to him. He will change.
Okay. Deepa, you ready?
Yes, ma'am. My voice is clear only, no, ma'am?
Yeah, nice. No problem. Next time we should put like how the ISRO fellows and all do countdown, no?
Ah.
Playing some music before.
Hi, Pushpa.
Who is that?
Pushpa madam
Pushpa,
is saying
Pushpa.
hi to speaker.
Hi, how are you?
Ma'am, sir is changing his computer.
Okay, okay.
Something. You'll have to
So
wait
he
for a minute.
changed. I saw his. I saw. That's why.
No, he's here, but he's, I think, changing his computer.
I'm still there.
Laptop.
I'm just changing the computer so that there is less of echo. Meeting ID.
After this, people have immediately call.
Maybe he can check his slides in the new computer.
That is done. That is done. Only it is connecting. Connecting. Yeah? Sir, can you see me now? Sir, can you see me now? Sir, can you see Madam. Hello.
Sir is seen from.
Madam, I have shifted to another computer. You have to make this host so that I can share the screen.
No, which is the computer, sir?
Elangovan NIE. Elangovan NIE Chennai edition.
Okay,
With
fine. 1 minute.
video, ma'am. With video.
Yeah, I can change. Sir, the thing is,
Sir, face is not seen closer.
your face is not coming. It is very far off.
We are zooming in.
Okay, the other thing account you need to log out, sir. It is echoing because of that.
Okay.
Yeah, now sir is seen closer. Sir
No, that is because he's
I'm
now
logging
coming
out from this. Now it is okay. No?
You can leave meeting, sir.
Now, new two portals are seen.
That's what. Now they have left. Okay.
There are two are there.
Sir, you want to check slides?
Yeah.
Okay, you can try now.
Have you made me host?
I have made you presenter, but before
Okay,
that, this is not starting of the program. Before that, Dean will introduce you and all. This is just
First let us just let us see the slides.
Sir, you are the presenter. You can check your slides now, sir.
Share the screen. Share.
Sir is trying to share. Your computer screen is seen, sir. Can you show the slides, sir, here?
Mr. Srinivasan, I'd suggest that you open the PowerPoint, minimize it and keep, then go and click share button.
Yes, open the
You are
PowerPoint,
seeing
ma'am.
no, I'm not seeing the PowerPoint. I'm seeing only the folder, lecture FFF folder.
PowerPoint is behind that, ma'am. They have to just minimize these two screens. Then the PowerPoint will be visible.
Sir, can you minimize this?
Minimize
screen?
these two screens, Cisco big screen and this folder screen, both these things.
Yeah. Actually, sir, this is your Cisco screen, sir, not the other one. Now it is seen, sir. Now it is seen.
Yeah, sir. Now
Correct.
full screen.
Correct, sir. Very good. All well, sir. Fine.
Shall we start, sir?
Yeah, Deepa, you start, Deepa.
Okay, sir.
I'll make Dean sir as a host.
Good afternoon, everyone. Dear colleagues and friends, now we are in this Forefront of Future Frontiers webinar, popular by the name FFF webinar. It has become popular across the nation. A medical webinar series to translate novel scientific innovations and integrate them into practice. It's been organized by Scientific Society of Arapadavidu Medical College, Vinayaga Missions Research Foundation. After completing 9 series successfully, now we are in series number 10 with the theme as quality and safety in healthcare, putting patient safety first. We have crossed the golden jubilee, which has got a very huge response. That I think that's why our YouTube also got is going near the silver button because of our 50th golden jubilee webinar we had. And last week we had a special Q&A session with that international speaker we had during our golden jubilee with a great response. Today we are at episode number 57 of series 10 with a novel topic, bioinformatics and data science in present day drug discovery, topic of importance in future. We have a resource person today from a reputed institute, Dr. Subarna Roy, scientist from ICMR, National Institute of Epidemiology, Chennai. Now I'd like to request our beloved Dean, Dr. P.F. Kotu sir, to give the opening remarks in the scientific session and also to introduce our speaker today. Over to you, sir. Sir is muted, sir. You are muted, sir. Please unmute yourself. Dean, sir, you are muted, sir.
He's still muted, I think.
Dean, sir, you are muted, sir. Please unmute yourself, sir.
No, it was my turn to get disconnected, connected.
Okay, sir.
Okay. Anyway.
Now we are able to hear you, sir.
Oh, yeah.
Yes.
Very good afternoon to all the participants and the resource person. I bring greetings from our Arapadavidu Medical College, Pondicherry, on behalf of our management and behalf of our university. It is my honor to welcome you all for this 57th episode of our prestigious FFF webinar virtual platform. And today's speaker, of course, Dr. Subarna is my good friend of mine from my Belgaum days. And he will be speaking on a very relevant topic under this, the quality and safety and health theme. That is the theme of this 10th series. And, drug delivery system, we have seen, from our student days getting evolved or getting unprecedented progression from a simple oral, intramuscular, subcutaneous to various sophisticated drug delivery systems. Now we have reached something known as a smart drug delivery system, or on a nanotechnology platform or a nanobiotechnology platform, and it is what is known as a stimulus sensitive drug delivery system. That means targeted place and a defined precise dose without affecting anything around. Now, suppose you are targeting a cancer cells, it should be only to the cancer cells, all the other cells surrounding should not be affected. This is what we understand. Zone, which I will be sharing with you in just a couple of minutes, and he will be sharing his experience on this topic. So I once again welcome him on behalf of our Honorable Chancellor, Board of Management, University for this platform. And already Deepa has said, this is a platform has earned quite a lot of credibility, and we talked about YouTube. It is on a national front, we're heading towards, I'll say, a centenary, since the 100th episode. Certainly, we're going to do it. And with this, some thoughts, let me introduce
Deepa, I think sir is last.
Yes, ma'am. Sir is maybe because of internet problem. Sir has left. Sir is joining, ma'am.
Some issues, he's quitting and joining again. So in the meantime, we have some this thing for the participants. For the regular participants, I have shared the website link and also our e-certificate link. There are a lot of queries regarding e-certificate. Now it is much less, but still some of the new people keep asking. You need to use your Cisco ID because we actually have tagged the e-certificate to the ID with which you are joining the this particular meeting. So we will pull out the attendance from Cisco and then we will be using that to give you the e-certificates. So please use this, remember this ID from which you are joining and use this as your username to download the e-certificate. We are just waiting for sir to join. I think that's why it is taking time. But sir, what shall we do? Wait.
You can talk to him over the phone and see if it is a major issue, then you can do the introduction.
Okay, sir.
Some connection problem, something happened, I think.
Sir is joining back right now.
Oh, okay.
I think it might be better that we put everybody on mute, no? Phones. There's some
See,
other
this
disturbance.
announcements.
You can
Meanwhile,
finish all that.
before sir will join, let me few announcements for the participants. The program is recorded for academic purpose, so it will be available in our AVMC YouTube. Anytime you can go to that. And I request all the participants to kindly mute yourself for clear audio visual thing. And please post your queries in the chat box. The Q&A session at the end, for the Q&A session at the end of the this one program.
Is it a computer issue?
It's not computer, some internet issue, I think.
There is one more system we can join from there.
Dr. Mahalakshmi, are you able to hear me now?
Yes, sir.
Yes, sir. Yes. We can hear.
Very sorry, I got totally disconnected. I don't know why. They have not yet resolved the problem, I think, in that we have WebEx. It is telling quit and quit. It's telling, okay, make me host.
Yes, sir. 1 second.
So I was telling, did I introduce the Subarna or no? I don't know when I got
Not yet,
disconnected.
sir. Not yet.
Okay.
Please go ahead, sir.
Yeah. I'll share that. Oh, yeah. Again, once again, a very good afternoon, and my apologies for getting interrupted like this. Anyway, it was a bad day today for all of us, and everybody had this kind of a problem. And I said, we're heading towards the diamond later on, 100 episode, and this have earned quite a lot of credibility. I was happy to hear that we have earned the silver button in the YouTube as well. The Professor Mahalakshmi was telling. Now, today's speaker, Dr. Subarna Roy, is a good friend of mine, I said, from Belgaum RMRC days, and he is a senior scientist. Now, presently, he recently got transferred to National Institute of Epidemiology, prestigious at Chennai. And this is his experience. He was working practically all major ICMR centers. He has spent his stint in Port Blair, Andaman Nicobar Islands, then he worked in Belgaum, and he has been instrumental in this Government of India, the Department of Health Research Project, the establishment of Model Rural Health Research Unit, Raichur. Raichur is in Karnataka, in the north western part of Karnataka, wherein he has been instrumental as a nodal officer in establishing this unit. And one thing, why this slide, even though it's difficult to read for all of you, one message what it clearly gives is all these courses and credentials what he has earned when the COVID struck, see, from 2021, from prestigious institutions across the globe, St. Peter's University, like University of Carolina, UK, John Hopkins University, when many of us wasted our time, and he utilized this time in getting these various courses which were available. It shows the commitment, dedication, interest, and passion. Then only a person can do this. Everything requires effort, everything requires interest, everything requires time. This is the message I can give, rather than all the reading all these details what he has done during this period. Yeah, and He has many awards and prizes to his credit from the prestigious regulating bodies, ICMR, NAMS, and from the University of Burdwan. He's a gold medalist from the prestigious Burdwan University. And he has this Major General Sahib Award from ICMR, Mishra Memorial Award from National Academy of Medical Science, and his Medical Innovation Fund Award. And he had, by virtue of his experience, by virtue of his passion, dedication, he was instrumental, because I know all these things personally. All the major medical institutions I have in Belgaum, Hubli Dharwad, then Dharwad is another college. All these places he has been the members of the Academic Council, Research Advisory Committee, and he was the Ethics Committee Chairman of our prestigious university, Kale University. Then he was a Ethical Committee Chairperson of the Belgaum Institute of Medical Science, even the age-old Karnataka Institute of Medical Science. So that shows, his commitment towards the profession. And he's trained in the sequence characterization of untyped strains of rotavirus, by the ICMR. He was deputed by ICMR to the CDC Atlanta, USA. And he is part of a 4-member scientific delegation which visited Japan for the study of the antimicrobial resistance. And he has been a reviewer and a peer reviewer of many journals, including the Lancet for Infectious Diseases. That's a wonderful thing. And his major contributions are the detection of investigation, first ever subsequent outbreaks of cholera in Andaman Nicobar Islands, and first civil government servant to be deputed to Car Nicobar Island. You know this Car Nicobar Island is prohibited; nobody else can visit there. Very much, lot of restrictions were there. When tsunami hit, those tribals, they don't receive any foreigner, anybody, not foreigner means any other persons other than these tribal people. So they must really admire Dr. Subarna for having ventured to go there, and that too for the relief operation. And he has developed the random amplified target sequence, a RATS PCR, rapid method of genotyping, and he had identified a clone, Gymnema sylvestre, difficult to read. And that is a much traded traditional medicinal plant used in the treatment of diabetes. Then the other, his work on rotavirus, and getting included in the universal immunization program, it was studied in the northern part of my state, and Maharashtra as well, neighboring Maharashtra. So the burden of the rotavirus was studied and inputs were given to this, and it was included in the immunization program. And he's the first to establish the human pericovirus and sallivirus as a casual agents of children diarrhea. A lot of work on that, and the diphtheria. And I already mentioned about a nodal officer for the DHR recognized establishment of a model rural health research unit at Raichur. It is his baby. And he has been a PhD guide for the Pondicherry University, KLE University, Symbiosis University, Pune, Shivaji University, Kolhapur, and University of Madras as well. And one thing, quite mind-boggling is he has extramural grants as a principal investigator to the tune of 72.28 crores. So that will reflect the involvement of Dr. Subarna in the research, funded research. And a total extramural grant as a co-principal investigator is another 2.17 crores. So this will just give a message to all of us, if a person minds what all he can do. And his publications are over 89 depth chapters. He has got 2 patents, and his indices are H index and I index are here. I index are 43. He has nearing 1,500 citations. So this is in try to brief. Of course, his publications and all. I didn't dare to go into the details of that because otherwise that will occupy the whole of this webinar time itself. Already we got some 20 minutes in this. So with these few introductions, and I'll request Dr. Deepa to conduct the further proceedings.
Thank you, sir, for such a brief introduction. His CV was amazing. Now we'll move on to the main subject of the day, Dr. Subarna Roy, sir. Over to you, sir. You will be able to share your screen now.
Yeah. Thank you very much. I hope I'm audible.
Yes, sir. You are audible, sir. Your voice is clear.
And screen is shared.
Yes, sir.
Is my screen shared, visible?
No, sir. Not yet, sir. I will. You are able to share, no, sir?
Yeah.
You'll be able. You'll be able to share, sir. I have made you the presenter.
Okay.
Your computer screen is visible. Your slides are visible, sir. Please make it full screen. Yes, sir. Yes, sir.
Yeah.
The screen is yours, sir. Go ahead. Thank you, sir.
Thank you very much. And at the onset, let me thank Professor Kotur and Dr. Mahalakshmi, Dr. Deepa, the faculty for giving me this opportunity to speak to you. And as I can understand, this FFF program had been a big success. And I am really awed with the previous presenters who had actually been given this opportunity to speak. And I find myself in that elite group is a really big honor for me. Thank you once again, and I'll be indebted to you all for giving me this opportunity. I only hope that I take more time than Professor Kotur has taken to describe my work. Honestly, I am more comfortable on the other side than on this side. Because I love to be a student and every day is a learning experience for me. I am not an expert in any subject, but I tried to be kind of a jack of many things. And I would like to share some of the new things that have actually come with the progress of informatics sciences, information technology, high-end computing, which we actually need to know in our daily lives, and we are from a very wide spectrum of health sciences working in various areas. And I hope that this generic talk will be useful to all of you. And with this, what I will do is that I will speak mostly on the classical drug discovery pathway and the reverse pharmacology pathway of the drug discovery, particularly from natural leads, where actually I have mostly worked on, followed by the use of bioinformatics as tools in the drug discovery processes, computer-aided drug discovery and designing, and the strategies which are usually taken for efficient drug discovery, the commonly used databases and software tools that are available at our repertoire, and the next-gen informatics and the big data biology that we are living in, and a few comments on the future perspectives. So, as we all know that the classical drug discovery pathway is a very cumbersome and a very long-drawn process involving a lot of efforts, and that requires a lot of expertise and resources. So where actually we start with a known target, where we have a disease, where we have a pathway, or where we have a molecule which is targeted. And what we don't know is the pharmacokinetics, the suitability of the test drugs, their safety, efficacy. And then we try to understand them. We do the bioavailability studies, we do classical pharmacokinetic studies, the safety, the efficacy. And so therefore we start from a compound library, a pool of targets-based drugs or lead or probable chemical entities, which leads us to the structural activity or optimization, and then the preclinical research, and then followed by clinical trials, and then we have a successful drug. So this is the classical drug discovery pathway that most of us know about. But as I said, that it comes with its own downside, which is actually a very costly affair and as well as a very time-consuming affair. And ultimately, when we start with
Dr. Roy, can I interrupt for a minute, please?
Yeah.
There is a lot of messages on the chat box that there is echo and disturbance in the voice. Can you use a headphone, please?
I don't have a headphone here, but what I will do is perhaps switch off the other connections.
Ma'am, but with headphone, it is with my headset, it is clear only, ma'am. This voice.
Yeah, but
but
not
messages
everyone
have
is
come.
using headset.
Yes,
No,
yes,
Deepa.
ma'am. That's why many messages have come. Audio is not.
Am I clear now? Am I
No,
clear?
sir.
Still
No,
echo
sir.
is coming?
Yes, sir.
Still it's echoing?
Yes, sir.
No. Hey, not echoing.
Echoing, sir.
If many people are having echo, so there might be some issue. Okay, I will call the technical person.
Sir, you're using two laptops nearby connected, sir?
No, I have switched off the other one. I switched off the other one.
Okay, sir, we'll continue, sir, and by then I think your technical team can come and
Yeah, I will
sort out.
I will just see if I can manage here. Just a minute, give me a time. Yeah, I will proceed with till the technical team comes.
Yeah, please, sir. Thank you. Thank you very much.
So now things are that we are now moving from what we call as new chemical entities to new therapeutic entities. So which essentially means that instead of starting with single molecules, we are now looking at drugs which are combination of various molecules. And so this is a new trend which has come, and these are actually taking over from various modern, from various existing systems. And a major lead actually is obtained from the traditional systems of medicine, be it in India or abroad. And what is generally done is a reverse pharmacology approach is adopted, where the safety and efficacy of these known substances or these entities are known, whereas the target remains unknown. For example, we have clinical evidence of effective and safe use of a natural medicine. So echo is over. So is the echo still there?
There's a slight amount is still there.
This is switched off.
It's manageable, I think.
If it is manageable, then we'll proceed.
Yes, sir. Please proceed, sir. Please proceed, sir.
Okay. So where we have clinical evidence of safety and efficacious use of a particular substance or a mixture of several drugs which are used in traditional medicines, and then we carry out some preclinical research, particularly for understanding the mechanism of action, and then from there we can develop actually individual molecules or maybe a combination of several molecules. So this is the reverse pharmacology approach which is generally been used, and now this is one of the approaches which is used for repurposing of drugs also. As you know that most of the drugs that are being tested or evaluated for the SARS-CoV-2 right now are also, most of them are repurposed from various other drugs. So that also uses this kind of approach. And there were several success stories from these approaches. For example, the artemisinin from Artemisia annua for the used in malaria. Ralphia alkaloids, which are used from Ralphia serpentina, used in hypertension, Sonolens for vitiligo. Then you have Parkinson disease, you have Mucuna purpurea, and the picorosides for hepatoprotectant activity, curcumin for anti-inflammatory and anti-cancer activities. Then you have steroidal lactones, the immunomodulators, Plaxidaxel from Taxus, which is used for cancer treatment, and Silymarin from Silybum medianum for the treatment of liver diseases. So like that, there are several success stories which have actually originated from the traditional use or from the traditional systems of medicine across the world. And therefore, natural drug industry, the ones which are actually based on natural products, there have been a renewed interest in that. And it is estimated that in the year 2005 to 6, perhaps the trade in these medicinal plants or the natural products were about 3.2 lakh metric tons. And the herbal industry itself is around 2 lakh metric tons of use, whereas you have rural households, you have exports, and around 960 medicinal plant taxa in India are traded. And among that, a priority list has been made by the National Medicinal Plants Board, which has 176 medicinal plant species in that. And the trade of that is more than 100 million metric tons. The resurgence of natural products or nutraceuticals or phytoconstituents have actually increased over the last several years or decades, and it has actually affected more than 80% of the world's population, mostly for the people who have been developing in the who have been living in the developing countries, and they rely mostly on the natural products as their primary healthcare. Mostly the access to modern medicines also is sometimes missing for them, and it is often viewed as an integral part of the culture of those people. Some of the reasons behind are the side effects or adverse events associated with modern drugs, problems with drug-resistant microorganisms which are emerging, the new diseases, emerging diseases like chikungunya, Ebola, the SARS-CoV-2, where no suitable medicines are available as of now, and the need for effective drug for chronic diseases or diseases that are difficult to treat with modern medicines, cancers, cardiovascular diseases, other lifestyle diseases, diabetes, and also something to do with the socio-economical and cultural aspects of these people. So now, as I said in the very beginning, the entire process of drug discovery is a very cumbersome process. It involves time and cost. Now, to reduce that, what is generally done is to reduce cost. People have undertaken simulation, computer-aided designings. To increase the data, people have tried to multiplex, do many things together, or high throughput, increase the throughput of the outputs. To save time, people have started doing things parallelly at one time. And to minimize the human errors, people have automated things. But one must be able to generate and cope with this large amount of data that is generated in a very small amount of time. And therefore, we must have trust in the process that actually has generated this data. So we are doing simulation, we are doing automation, we are parallelizing, we are doing multiplexing and high throughput output we are getting, but we must have trust in that system that is actually generating this kind of data. So what are the bioinformatics tools that are used for simulation? So these are some of the tools that are used in drug discovery process, such as for identification of the target disease, for screening of interesting compounds which can be used to target these diseases, detection of molecular basis for the disease in question, molecular basis of action of these compounds on the various targets, the rationalization of the drug design techniques. So drugs can be designed and modified. So I will come to that in the next 1 or 2 slides. Refinement of the compounds based on their efficacy or side effects. Again, I will come to it. So drugs can be modified based on the efficacy that is obtained or the adverse event that they may produce. And QSR studies, that is quantification of the structure activity relationship between the drug and the target. And determination of physical properties, that is the solubility of the molecules, so something like that. And of course, finally, the testing of the drug itself. So identification of the disease and the targets in the compound of these compounds. First, the disease to be targeted is identified. So all that what is known about that particular disease, the structures of the key metabolites or the players in that particular disease pathway is determined. Then the existing molecules, those that are hypothesized for the remedies are identified and their structures are determined. It is also important to look at similar afflictions and their known treatments. So to identify potential drugs, we generally look at similarities. If their target has got a similar target in something else, then we generally will look for those solutions that are there that targets that related disease or related compound. So, but then we must understand that target identification alone is not sufficient to achieve a successful treatment for the disease. We need to find real drugs for that. And for doing that, bioinformatic methods like docking are usually used to virtually screen through a lot of compounds that bind and inhibit a particular protein or a particular molecule. This is done in computers in silico, and it is just like to find a key to a particular lock which fits in well. There are situations where there can be multiple locks and multiple keys. So those kind of situations also need to be taken into consideration. The drug must influence the target protein in such a way that it does not interfere with the normal metabolism of the patient. What Professor Kotur was talking in the very beginning is that it should not harm the body itself. It should only target the pathogen or it target the disease molecule. At most times, the compounds that are likely to have some activity against the disease are screened. Not exactly the one which has very good activity or whatever. It can be somewhat related, likely to have some activity. And then if they are marginally useful, then their usefulness can be increased by doing minor modifications in those particular molecules. And these molecules therefore provide as a starting point for the refinement in the due process of drug discovery. So for detection of the molecular basis of the action of these drugs, it is generally known that a drug must bind to a particular spot on a particular protein or a nucleotide. Then the drug can be tailor-made to bind at that particular site. It is often modeled computationally using a variety of techniques and using various softwares. The primary way of determining what compound will be tested computationally is generally determined by two ways. One, if the researcher understands the molecular interactions very well, so through his own experience or his understanding, he will try to find out which drug is likely to work against a particular protein or a particular target very well. So he can have the choice on that. Or secondly, by a brute force method, again by high throughput screening, where you generally screen a large number of compounds that are available from a particular database against your particular target and then find out whichever works most. So this is the brute force method. Then you can refine the compounds if you get some likely activity or some activity. So this refining can be done by refining the molecular structure to give a better drug activity and fewer side effects. This is done both in the wet laboratory as well as by computationally examining the molecular structures to determine which aspects or which domains are responsible for both the drug activity and the side effects. Those domains which are responsible for the side effects are generally removed or modified. Similarly, those domains which are useful to bind to the target are modified to give it a very good fit. The drugs are then modified structurally to achieve the desired activities. And for this purpose, computer-aided drug designing a specialized discipline that uses computational methods to simulate drug receptor reactions are generally used. Therefore, CAD methods are heavily dependent on bioinformatic tools and applications and the databases. So therefore, there is a lot of overlap between CAD research and bioinformatics. So bioinformatics actually supports CAD research. Pharmaceutical companies are always trying new ways to develop new drugs, new vaccines, new novel compounds. One of the methods that they use is generally called VHTS, or virtual high throughput screening. In this, the protein targets are screened against a database of small molecules, and nowadays often large molecules also, to find out which of them binds strongly to the target. If there is a hit with a particular compound, that can be extracted from the database for further testing. And with today's computational resources, several million compounds can be screened in a very few days using sufficiently large computers or clustered computers. What we generally do, even small computers, we generally link them together and cluster them so that all the CPUs work together for a particular work. And therefore, the computing power of the computers increase. Pursuing handful of promising leads for further development can save researchers considerable time and expenses. So bioinformatics support to CAD. In this, the CAD research is often knows the genetic sequences of multiple organisms or amino acids of proteins of several species. So in this, we come to know the targets of various organisms or multiple organisms together. And it is very useful to determine how similar or dissimilar they are based on their gene or protein sequences. Now, classic example is about this variants of concern or variants of interest that we are seeing in the SARS-CoV-2. So this is exactly what we are trying to find out, how similar or how dissimilar these various strains are against each other, and how the vaccines affect on them, or how the drugs will actually alter the efficacy for them of this against these particular strains. Now, with this information, one can infer about the evolutionary relationship between the organisms and search for similar bioinformatic databases and find related species to those under investigation. If you can remember, when the drugs started against SARS-CoV-2, then people were looking at those drugs that were actually made for the original SARS or MERS when they were seen earlier decades back. So these are kind of things that look for similarity, both with the target as well with the probable drug that you are looking for. There are many bioinformatics sequences or analysis tools that can be used to determine the level of this similarity indexes. Homology modeling. Homology modeling is an important aspect in CAD research as well as in bioinformatics. The most important challenge is determination of the 3D structures of the proteins of the targets or even the drug molecules. Most of the drug targets are proteins, and therefore a knowledge of the three-dimensional structure is of immense importance. And it is estimated that the human body alone has about 5 lakh to 10 lakh proteins. But the 3D structure of only a small fraction of them is known. However, from the related 3D structure available of the proteins, the other 3D structures of those proteins which are not known can be modeled using computers. In 3D homology modeling, the amino acid sequences of known proteins, in this case the target, is unknown, and the 3D structure of a related protein or the target or template is known. And bioinformatics softwares are used as tools, and then the 3D structure of the unknown target or the specific protein is generally determined by using the template structure. One of the used bioinformatic resources, a modeler, which is a tool in homology modeling, as well as Swiss model repository, which is a database of protein structures which are created with this homology modeler. This is the screenshot of the modeler and the Swiss model database. These are available, and if you know the sequences of the amino acids, you put them together, and then you will find the the structure. If the structures are available, you will get the 3 structures there. If you don't get the structures, you can have the option of modeling the structure yourself using these tools, using these softwares. Autodoc and innovations are again some of the resources that are used in similar studies, where you can actually get model 3D structure and you can do the modeling itself. Another approach which is used in this connection is the subtractive genomic approach. In this, to find putative new drug targets and new drug discovery, what we do is that the proteome consisting of all the known proteins of a pathogen is retrieved from the database. So you get to know all the proteins or list them out from the database, whatever is known about the particular pathogen. Now, parallels or duplicates from that list is removed. There are resources how to remove it, like CD-HIT web server, which contains a percentage threshold with which you can actually remove a subset of these proteins, which are very similar or are very similar to each other. Then you can do a BLAST-P, which is performed against a human proteome to identify those proteins that are not identical to human proteins. So essentially, you are again removing those proteins from the pathogen, which are already known to exist in the humans also. And then the proteins are generally subjected to what we call as the DEG BLAST, or the database of essential genes and genomes, to find those proteins that are essential for the pathogen. Without those proteins, the pathogen cannot survive. So those essential proteins are chosen for the target. And now you remember, we have already subtracted it from the human. So those proteins will not be available in the human databases. Then there are resources and softwares which will actually give you the subcellular locations of these particular proteins also. Then the docking studies are done in silico or in computers, and then we can actually identify the leads of the known substances with which the activity of the pathogen can be established and the likely mode of action can be done. So therefore, this is the best way to identify or target proteins in the pathogen, and then select your probable drugs which you can use to target them. There are several databases of natural products which are related to this chemical constituents and this kind of work. The National Medicines Comprehensive Database, HerbMed, Nube. I will come to a few of them. I'll show you the screenshot. These are the screenshots of some of the bioactives databases. Even PD, Universal Natural Product Database, is one where you can actually search this natural product database.
Mail to
Yes, sir.
and put it back here.
Dr. Duke's phytochemical and ethnobotanical database, again on traditional systems. So you will find a lot of phytochemicals listed in that database. And then you will find some of the resources in which you can actually relate them to your particular study that you want to do. If you look at chemistry, even PD will have some of the chemical properties that you would like to know,
Okay.
along with the ChemBL database and the drug bank. For disease targets, one of the best sites or best the database repository is BindingDB. From BindingDB, you will get quite a lot of targets for various diseases. ChemBL also will give you some kind of information on various chemicals or various chemical entities that can be disease targets. SwissDoc, again, you can do docking as well as you can identify targets with that. There are several others. Uniprot is another one. Now, disease target association, once you try to associate them together, then therapeutic targets database is one of the best used resource where you will have disease and target pairs combinations. Human protein atlas also will give you some information on the human component of it. And disease and gene annotations, if you want to relate more with the genes, then you can get from there. And if you want to develop systems biology or network development with how individual compounds and individual targets are working together in a system, then KEGG pathway is one of the most used resource. David is also one of the bioinformatics resources which can be used for developing networks and system biology. And string is also nowadays used quite often for this kind of activities. The Chinese have actually done quite a lot in these areas, and the Chinese traditional medicines, and they have a big database and repository of various probable medicinal leads from which modern drugs can be developed. And this Chinese traditional medicine database is a comprehensive database where constituents from 352 herbs and animal products and minerals are listed, and contains more than 37,000 traditional medicine compounds, and provides comprehensive ingredient structure and information for each TCM. And you can search these with chemical names, the traditional name or molecular properties, their structure, or even a basic search and advanced search on that. Then you have something different, very similar to that, but a little different from that, which is the traditional Chinese medicine integrated database. Here again, you have information about 8,000 herbs and around 25,000 ingredients and around 6,800 drugs and almost 17,000 disease targets. And it has got information about prescriptions also about 46,000 or 47,000. And the herbal ingredients target database also is one which I mentioned earlier. And this is a comprehensive and fully curated database of protein targets for FDA approved drugs, as well as some promising precursors. It contains about 1,300 known protein targets and around information taken from around 3,200 literature that cover about 856 active compounds and about 1,300 reputed Chinese herbs. The Universal Natural Product Database, UNPD commonly known, has got almost 2.3 lakh molecules listed, and it allows template-based search for molecules and for natural sources. Supernatural 2 also is a very similar resource which can be used, and it contains around 325,000 listings of natural compounds with their 2D structures, not 3D structures, but their physicochemical properties, predicted toxicity, and even it lists out potential vendors from whom you can actually get them. So there are several other databases also. Nubbe database is more of Brazilian biodiversity. So there are several resources or databases from various countries, but the ones that I listed are more commonly used in various countries across the world. And most of them are freely available, and you can actually search them on your disease of your interest and your targets. And even Natural Product Alert also is one which can be used for related research activities. And it contains around 2 lakh scientific papers and reviews on particularly the ethnomedicinal information about the natural products. And I'm not going into the detail of it, but then there are quite good information which are available in this particular database. One example is the TKDL, the Traditional Knowledge Digital Library, which, if you remember, was launched actually to prevent patenting of the Indian traditional knowledge, particularly from the systems like the basmati rice, the kurkuma langa, the haldi. And these are the kind of information that is available in this database, and this cannot be patented by anybody outside the country. IUSoft is something which is based on the Ayurveda practices in India. Again, this is a small one, but still something which is worth mentioning, particularly on Indian system of medicine, the Ayurveda. And what I have, this is a very busy slide, but what we generally do is that we have made a kind of a workflow for what we call as herbal informatics. I am not an informatician. I am neither computer science expert, but I am a biologist. So I know that I am working on this particular disease, and these are the kind of drugs which are used in the traditional medicines or even in modern medicine or in the pharmaceutical industry. And then I would like to do studies on these particular drugs, whether they are really effective or whether they are not, or what are the phytoconstituents that may be taken for further drug discovery or identifying novel molecules. So what we do is that we combine the knowledge of traditional medicine as well as high-end bioinformatics. Say, for example, here I have given the example of diabetes mellitus, where we have listed out a lot of various drugs or polyherbal formulations which are in the market, which can be used for managing diabetes. And then we mind all the pharmaceutical compounds on their canonical structure and their smileys which are there in those particular products which are there in the market. And then from there, we have listed out the individual compounds. And then with these individual compounds, we have used KEGG pathway to understand where actually in the metabolism of diabetes that actually these compounds work. And then once these are known, then these are mapped in what we call the in a systems biology approach. And then we found that around 82 enriched molecular pathways are affected by a large number of these compounds which are available in this particular marketed products. And we identified 21 protein targets which are modulating 11 pathways in this diabetes mellitus. And then from there, we generate a compound target pathway network. And then from there, we do a docking studies individually to find out whether they have any or they may possess any possible adverse events or adverse effects. So, and then whichever pass, then those kind of compounds can be even taken into preclinical trials with animal studies. And then if we get good results from them, that can be taken into clinical research for developing novel compounds or novel drugs for management of diabetes. So this is, I just gave you an example with diabetes, but this can be done for almost any disease of your interest. So these are even small publications that can be made on purely computational studies. And why I'm telling now is that with the SARS-CoV-2 around, you will find that we did not have much time to actually develop new drugs or solutions to tackle this disease. So a lot of bioinformatics studies has been undertaken, and every day large amount of data is being generated, and these are the kind of simulation studies that are being done with existing drugs, with existing traditional knowledge, and then put together to find out which works best, and then taken into the wet lab laboratories or into clinical research mode to find out the best possible solutions. Now there is a information boom, the big data biology. We have a lot of data today. Earlier, most of the research used to be curiosity driven. So we as scientists or as faculty or as students used to be interested in something, some research question to come, and we used to do research and then find out the solutions or answers to that. Then came the age when actually it became a objective driven research. You know the expected outcome. There is a call for proposal that I want to do this, or we want to achieve this. This is my final thing. I want to have a drug for this particular disease, and you develop them. So then this became the targeted research. Now we are entering into an area when actually we are going into data-driven research. So we have a tremendous amount of data. We don't know what we are going to do with that data, but there you have to find research questions from within the data and then find the best possible solutions from that. So now research is being driven by data. So that is why data is so important in this particular world. And it is, if you look at the growth of biological databases over the years, as you can see in this graph, how much the biological databases have grown over the years from say 2006 to present 2016 or even 2020. The data increase has been extremely high as far as biological databases are concerned. If you look at published genomes, this slide also is quite an old slide, but still there are more than 1,100 published genomes. It is much more. And this data collected is now at a much faster rate. If you can see the submissions, so it has just taken a steep curve. So the enormous amount or enormous rate in which the data is being generated is extremely high. So this is a database of SNP single nucleotide polymorphisms. So and particularly for genomics. So data are actually increasing at a very fast pace. Now, if you look at the published articles, Medline published articles, so the number of research articles also is increasing tremendously. Earlier, publication itself used to take so much of time. Now you have even BioRx and all, where even before being peer reviewed, you are getting access to publications which may be published later on after the peer review process is over. Similarly, the PubMed open access papers also have started increasing, and most of the COVID research, as you can see, are now free to access. So access to research papers are also increasing tremendously. And if you look at open access growth journals from Elsevier, then you can see by 2020 how much the gray ones are actually the open access journals. So most of them are coming into open access, and people are actually having access to data which is being published. This plot is again a little reverse. Actually, it is from the recent to the past. So essentially the same thing which it says is that the percentage of what is being written nowadays and is getting available online. So earlier we used to write many things, but never used to get online access to it. But nowadays online access is also increasing, even other than research papers and scholarly articles. Now, if you look at the discipline-wise publications, again Elsevier data, if you can see the top few journals or the top few disciplines as such are mostly related to our health sciences. Multidisciplinary research, dentistry, veterinary, pharmacology, toxicology, biochemistry, genetics, molecular biology, medicine, agricultural and biological sciences, health professions. So these have actually dominated more than physics, astronomy, neuroscience, mathematics, and other disciplines. And if you look at the publications as such, then if you can see the bioinformatics publications have actually become much more these days than it used to be earlier. The red one is the bioinformatics publications, followed by the green one, which is on genomics, and then the blue one on proteomics, and then the light blue on computational proteomics. Now, once we are in today's world, a little bit on the future. If you can look at the present doubling time of the next gen whole genome sequences, only a few months. And if you look at the doubling time of data storage capacity, it is about 18 months. A simple example, say you have a 256 GB storage SIM, or rather a memory card. You will get the same card for double the storage at the same cost in around 18 months time. So the cost of storage or the doubling time of data storage is much more than actually the doubling time of the data. So that means data itself is increasing much more, and the storage capacity increases at a slower rate. So therefore, nowadays, if you look at the cost of even our emails and all also has increased. So people like Gmail and all, they are actually collecting data for collecting money for the storage of our data. So therefore, the storage cost is going to increase even more. So that is what I am telling, is that the data is increasing very much, but the data storage capacity is not increasing in that rate. And so this is going to be a challenge in the future. And the most of the for scientists, another challenge will be to make sense of the enormous amount of data that is currently being made available, and to utilize them. Even you will read every now and then data breach and your personal information is gone and all. I also don't know what they are going to do with the data. Perhaps they also don't know, but it is being sold in the what they call as some gray market or black market, something like that. So that is being sold. The data people are sold. So data is costly, and we have to make sense out of this data in a research perspective and then find out solutions from this data. So I will end with a small video of in 60 seconds, actually, you are sending 204 million emails, 300,000 logins to Facebook, 1.3 million to YouTube, 1,000 tweets in 60 seconds, 60 hours of video, 62 hours of music downloads, and all happening in 60 seconds. And when data become big data, and then we are not talking about bytes and exabytes, we are talking about the petabytes now. And the variety of unstructured data is increasing, and the velocity in which it is increasing is tremendous. So the big data is the next gen and the next wave of research, which we have to use as a big opportunity for us, and the time to act is now. So we have with us a lot of data, and a lot of data is going to come to us. I'm a biologist. I'm sure all of you are in various fields, but we have to use and may take advantage of this big data that is being generated and make the best use of them so that we can use it in our daily lives as well as for the betterment of the healthcare of the people of our society. Thank you.
Thank you, sir, Dr. Subarna Roy. I was wondering what this is all about. Actually, to be frank, 2 years ago, when we used to have novel methods of teaching in the classes. Myself, basically, I'm an obstetrician, so I gave a topic like anemia in pregnancy for my students, and I asked them, you find out some novel method how to combat anemia in India, because people are not taking tablets. What to be done? That time one student came up with an idea of asking me whether we have the natural medicine as drumstick leaves. I don't know the actual name which you have been mentioning till now, sir. So that can be used for with a nanotechnology he has found out, and that can be used, and with nanotechnology the medicine can be made with more absorption, because the problem with iron in India is absorption. So he was telling that one. I was remembering when I saw the title today, what he was mentioning. It was some 2 years ago. And then I went through that, and we tried to do something on research on that. Meanwhile, we have been stuck up with this COVID thing. And sir has started with a success story with the natural medicines. And then he went through this traditional herbs and how it is useful, and then he landed up with this with all the traditional herbs which is used, and with the big data what we are going to do with that. And so the future of big data, sir, has emphasized we are going through this digital India, and how to incorporate this traditional herbs into this data, and how it will be useful for a developing country like India. So thank you very much, sir. We look for any queries in the chat box. Meanwhile, Dr. Bhatt, sir, you want to say something?
Sir, very interesting talk. These sites which we have told, are they free or it's a paid sites? Can you hear my question, sir?
He has to unmute himself and speak. Dr. Roy, you have to unmute yourself.
Yeah. Am I audible now? Yes, sir. Very important question. Even I actually had been in such labs where we had no money, and I started using bioinformatics because I could not do much. Purchase of high-end chemicals and all was not possible. So that is how I started with bioinformatics. That does not cost much. So what I do is the most of the resources that I showed comes free, and you without spending a penny, you can actually do quite good amount of research using the freely available tools. At the most, sometimes some resources will ask for a .edu or .ac academic email IDs. So if you have this academic email IDs or .edu or .gov, sometimes you get much more access to this resources. Sometimes what my students do, they download this evaluation versions, and within 2-3 days they finish their work and then just delete it. So there are ways actually in which even without paying anything we can do quite some substantial work. But there are quite a good number of resources. Say for example, one Schrodinger is there. It is the kind of most useful tool which is used in bioinformatics. That is quite expensive. That is quite expensive, but there will be various laboratories which actually have it, and most of the people who have these softwares will actually welcome any student or any faculty coming there and getting things done.
Another question. This herbal medicine which we are using, most of the times they use multiple herbal things in one. So how to find out which one is really acting and in the disease? Because many times you don't find them using single medicine, they using multiple plant products like that.
Yes, sir.
Even if you take a single plant also, there may be 2-3 alkaloids. So how to separate that?
Even our experience is also like that. What we used to do in our wet labs is that we used to fractionate and then go by activity guided fractionation. But after some time, the activity to decrease. So as you rightly said, there might be 2-3 or more compounds which are actually acting in synergy for that particular activity. So now what happens is that with this, if you look at my the diabetes slide, what we have done is that we have put multiple compounds which are available in a particular plant in that KEGG pathway, and we have given the diabetes as the target disease. So whatever are the biochemical reactions which are associated with the metabolism of sugar, so there the interplay actually is made in computational modeling. So they will tell which are the molecules which are likely affecting this particular enzyme or that particular enzyme. So there will be multiple pathways. So multiple compounds having effect on multiple reactions. And once you put them together, so then 2, 3, 4, 6. So this is what is contributing to what we now call as system biology or network pharmacology, because no drug, even a single molecule, perhaps also will have some kind of effect in other pathways as well, which perhaps sometimes we did not know, and that is sometimes even a drawback of some of the modern medicines as well. So a network pharmacology or system pharmacology, if we are just trying to make at least a few networks, and then we will get a clearer picture about what is the path that it is following and whether it is a single compound or multiple compound which is affecting in multiple ways. But it's a complex thing, but we will only get to know only a small fraction of it. But as I said, the data is being generated so fast. So in 5-6 years from now, we will be knowing much more than what we know today.
Probably this is one of the main reasons why allopathic doctors are finding it difficult to understand Ayurveda or the Siddha system or other system, because we are giving one antibiotic, one sedative or one drug for a condition. So when we say that multiple things act, we are not able to understand how they act together and what is the issue.
Sir, honestly, we have to actually tell if this particular molecule is acting on this particular enzyme or this particular protein, and another molecule B is acting on this, and together it is acting on this. If we say like this, and then it is really understood, and even kind of evidence generated with some preclinical studies, then anybody will accept it.
Thank you, sir. We have one question from the chat box. Role of bioinformatics in vaccine development. Is it employed in COVID vaccination?
It is because all companies will be doing that first. I think that is the first. This is a very small step that I showed, and also keep in mind that this is ultimately a computer generated or a simulated study. It is just like artificial intelligence. The more data you put into it, the better it becomes. So it is in an evolving stage, and we tend to believe it. The more and more data comes into it, the more data is fed, the better it is going to be. So it is a beginning, but yes, most studies, including vaccine development, will happen with this.
Okay, sir. Thank you, sir. One more question from HOD Pharmacology, Dr. Ashokan, sir. What are chimeric molecules and how it is made by drug designing by using bioinformatics? Want to know about chimeric molecules.
Sir, please unmute, sir.
So,
sir. Good
as I
afternoon,
said,
sir. Sir,
yes, sir.
this is Dr. Ashokan. Actually, these enantiomers we are using for the pharmacology drugs which used recently for treating so many diseases. Enantiomers, they are all designed actually by these actually by the drug designing done by these bioinformatics. Like that, these chimeric molecules. Actually, now we have our in our body itself we have chimers like both right hand and left hand are the chimers. Like that, the drug molecule also made by drug designing by each molecule by chimers, and the number of molecules are coming by chimeric technology. And is it also helpful by the bioinformatics, which is helping to come out with all these chimeric molecules?
Right. Yes, sir. So, in fact, sir, you would know, you will be the better person actually to answer this
Yes,
question.
sir.
Because,
yes, sir. Because when I start studying drug designing, I am very much confused, and we should know more chemical knowledge, chemistry knowledge for studying that.
Exactly. So you require
structure activity relationship, with one drug. There's so many actually molecules are coming out. Sir, Vishnu, but sir asked one question that they asked this doubt, regarding the number of molecules present in extract. Which molecule is acting for the disease? We don't know. That as per evidence-based medicine, we are using only one drug, but they are telling so many molecules in that. That is also studied by the, I think, this bioinformatics as well as the drug designing technology is used.
Yes. And one thing for sure is that, as you said about the chimeric drugs, so you will normally tell your bioinformatician that this is my molecule and I want this to be added or this to be subtracted. So they are the people who will actually play with that system and then tell you. So
yes, sir. Yes, sir.
we are more
yes, sir,
into
that's
the
why.
part. So, so the informaticians and the computer scientists, they will be actually doing the modeling part, and they will be advising us on this.
Yes, sir. Actually, this tocilizumab, actually monoclonal antibody. There are a number of monoclonal antibodies coming for the treatment of COVID. New monoclonal antibodies also under trial. But these are also, I think, designed by drug designing and bioinformatics only, which are helpful and coming out with a new molecule with the number of monoclonal antibodies. Is it so, sir?
Yeah, of course. As I answered my previous question, is that there will be some bioinformatics studies definitely before actually undertaking even laboratory studies, because this does not cost much, and they will know what most likely it is going to do. So once that is done, then the laboratory studies will take place.
Okay, sir. Thank you. Actually, we have to elaborate the knowledge in drug designing and structure activity relationship in the chemistry knowledge also regarding the drugs. Then only we'll be able to know how this bioinformatics getting used in drug design.
Thank you, sir. Thank you for your.
One more question is there in the chat box.
What are IND and NDA, ma'am? Sir, Dr. Martin Ahmed Khan has asked what are IND and NDAs, which you have mentioned in your earlier slides. He wanted to.
IND and NDA.
Sir has mentioned in his first or second slide. So.
Go get back to the slide. In the first slide.
Yes, sir. In the earlier slides, I saw IND. I think that's what he is mentioning. Dr.
One
Martin?
of the very first slides,
Yes,
Deepa.
sir. Yes, ma'am.
Second or third.
Before the success story, that slide, sir. Before that slide.
Before the success story. I don't see any IND. I don't see any IND.
Okay.
Anyway, that may not be too relevant.
Okay, sir. Thank you, sir. Thank you for your enlightening lecture on such a novel topic, which was totally new for most of us, but still we gained good knowledge on that, because this is going to be our future, and still this is the present thing also, which is going on in the drug designing thing for everything. Like as you told, for any vaccination which is coming normally, any monoclonal antibody, everything, we need to know about this bioinformatics and drug designing, how it has been done, and we need to know about this data also. You have told many of this thing. So thank you, sir, once again for giving your time.
Thank you.
Thank you, sir.
Thank
So
you.
I thank all the participants. I thank our Dean, Dr. P.F. Kotur, sir, Dr. Vishnu Bhatt, sir, and other esteemed HODs and professors and I thank all the postgraduates and other people who are participants from other institutes who have joined today with us. Thank you once again. With this, we'll wind up with today's session. We have next week, once again, we have our next webinar on this same theme of Series 8, Quality and Safety in Patient Healthcare Services. Sir, we have Dean sir. Dean sir, you want to say something?
Okay. Deepa, the next week.
No, Deepa. I just wanted to say thank you to Subarna. That's it.
Okay, sir.
Thank you, sir. Thank you.
Okay, sir.
Next week's speaker is Dr. Jyoti Clara Michael. She is Director of Nursing associated with many hospitals, Max Hospital, Columbia, Asia and all. She is going to talk about how empowering healthcare teams is going to help in improvement of quality of care. She is Director of Nursing from these hospitals. Yeah, Deepa.
So we invite you for the same in the next week once again. We'll let us all meet once again the next week. Thank you once again to everyone. We'll wind up with this. This is Dr. Deepa, Member Secretary, Scientific, AVMC, signing off. Thank you.
Thank you.